All right, welcome back, everybody. It's 12:35, so I hope you guys had a good lunch break. I would like to introduce the next speaker. Next speaker is Roland Canard, and Roland is a professor of molecular genetics and the head of the molecular genetics department at the Erasmus MC in Rotterdam. In his lecture, Roland will discuss insights in DNA break metabolism, both in the mechanism and the clinical impact. So, Roland, the floor is yours as soon as you're able to.
All right, thank you very much. Let me see. Okay, so I really appreciate the opportunity to share some of our data with the members of the X-omics large-scale scientific infrastructure consortium. We work as a department on the mechanism of genome maintenance by DNA repair. And the two stories that I will share with you today have to do with a specific DNA repair pathway called homologous recombination and a specific protein that's important in that pathway called BRCA2. You know that BRCA2 is involved in breast cancer, so this is the first of the stories that I'm going to share with you, has to do with breast cancer. And breast cancer is a very heterogeneous disease. There are multiple treatment options. So for breast cancer, it's really important to select the patients early on for the proper treatment. And of course, we have classical ways of doing this, pathological analysis. So we have novel ways of doing this using DNA sequence information. But the direction we have chosen is to do something that we call functional pathology. So instead of taking a biopsy from a patient and fixing it for the classic pathological analysis, we try to get information, functional information from that biopsy as to the functioning of a specific biological pathway, and then predict an optimal treatment for these patients. And so what I'm going to tell you about is a functional assay for homologous recombination from viable tissue material from breast cancer patients that allows us to predict that these patients should respond very well to treatment with PARP inhibitors, which are specifically a precision medicine for tumors that are homologous recombination deficient. So the idea is that we actually test tumors on phenotype rather than genotype. And we have set up a pipeline from the clinic where we take biopsies from patients to the lab where we grow them in organotypic slices. And then we can test for function of a biological pathway. There are a number of tests that we can do. And for this particular talk, we are looking at DNA repair and then specifically homologous recombination. And if I explain the assay, I first have to take you to some details of the mechanism of homologous recombination. So homologous recombination is a pathway that can repair, among others, DNA double-strand breaks, and it does that in an accurate way because when one of the sister chromatids is damaged, it uses the intact identical sister chromatid in the cell as a repair template. There's processing at the break, and eventually a key protein in homologous recombination called Rad51 forms nucleoprotein filaments on the processed DNA end. And these filaments have an amazing property in that they can search the intact identical sister chromatid for places where the sequence is the same, and then pair with the sister chromatid so that it can be used as a repair template. Now you can see that the Rad51 accumulates at a high local concentration at these sites, and these sites can be seen by immunofluorescence. So for example, here you can see a cell that is proficient for this homologous recombination pathway, and at sites of DNA damage, the Rad51 protein accumulates in these bright foci. This requires a number of different proteins, including the BRCA2 protein. So if cells are deficient in homologous recombination, then these Rad51 foci do not form, and specifically in BRCA2 or BRCA1 mutated cells, these Rad51 foci don't form. And this is the basis of our assay. We assay whether material, live material from breast cancer patients can form these RAD51 foci. And if it doesn't, then it's homologous recombination deficient, and the patient should benefit from PARP inhibition treatment, whether or not it's a genetic BRCA mutant, right? It could be many other different mutations upstream that lead to the lack of RAD51 foci. So in that sense, it's a functional assay. So we take fresh tumor tissue from either primary tumors or from metastatic lesions. We take it to the lab, we irradiate to induce the breaks, and 2 hours later, the foci detection assay is finished, and you're either homologous recombination proficient or you're homologous recombination deficient. We have done this now for over 300 tumors, and we find that homologous recombination deficiency by the RECAP test, for a repair capacity test, is not that rare. So in 20% of primary tumors, they are RECAP negative, so homologous recombination deficient. In metastatic lesions, it's even higher. It's about 30%. And so this assay is now the basis for a clinical trial, for which I get to in a minute. But before that, I want to share some data with you about the sequencing of some of these HRD tumors. And it turns out that most of these tumors, in case of the primary tumors, are not due to germline BRCA mutations. They're due to somatic events that involve BRCA, mostly BRCA2 mutations, and hypermethylation of the BRCA1 promoter. For the metastatic lesions, most of them are not BRCA related. And in this large group here, we have been able to identify, for example, a PALB-B2 mutation, which is a protein that interacts with BRCA2 and is also important for its function. So that means that our assay picks up non-BRCA related homologous recombination deficient tumors. So the assay is now the basis for a phase 2 clinical trial, which is a multi-center, and it's sponsored by Pfizer. So patients with locally advanced breast cancer or metastatic breast cancer are eligible for the trial. We take the biopsy, we perform the RECAP test, and once they are recombination deficient, these patients are treated with tolazaptorib, a PARP inhibitor, and then we are correlating the clinical progression of these patients with the application of the drug. We're also sequencing 17 patients that are homologous recombination deficient and 17 patients that are homologous recombination proficient at the whole genome level to figure out if we can see any correlation between DNA-based or genomic scar-based methods for predicting homologous recombination deficiency and our functional assay to predict homologous recombination deficiency. The nice thing about our assay is that it gives you a real-time measure of homologous recombination deficiency. So, for example, we had 3 patients in which we had a sample taken before the start of treatment and when they showed progressive disease. So all these 3 patients were homologous recombination deficient, so there were no RAD51 foci. And once they came back in the clinic after treatment with DNA cross-linking agents or PARP inhibitor, and they showed progressive disease, then their tumor had a homologous recombination proficient phenotype. So it gives you sort of a real-time view of it, even though these tumors that are now homologous recombination proficient, of course, still have the historical scar in their genome of being homologous recombination deficient. And for one patient, we have been able to figure out what the mutation is. So this patient was homologous recombination negative, was put on carboplatin and a taxane and a PARP inhibitor. And when progressive disease occurred, we found that it had reverted to homologous recombination proficiency due to a secondary mutation in BRCA1 that restored a reading frame. So in conclusion of this first part of my presentation, we showed that we have a functional assay for homologous recombination capacity using viable tumor material of patients. This assay picks up homologous recombination deficient tumors at a rate of about 20% for primary tumors, breast cancer, 30% metastatic tumors. We can detect reversions, so we can have a sort of a real-time readout, And a big question that we would like to address is whether our functional assay correlates is in accordance with DNA-based tests, or whether we pick up different things, for example. And in addition, as I said, that breast cancer is a heterogeneous disease with many treatment options, we're also now expanding our assay to directly test for sensitivity to chemotherapeutics that these patients get. So for example, taxanes and anthracyclines, so that we can also work out a predictive assay for those, which is very important specifically in the case of anthracyclines, because it's a drug that has lots of toxicity to the heart. Many breast cancer patients get it, but only a quarter of the patients that get it have benefit from it. So it'd be very important to be able to predict that in the future. Okay, and now my screen is going, now I'm losing my presentation and I don't know where it went. So let me see. We're back. Good. So this is the first part of the presentation. Here's the team. Dick van Gent is the researcher that is in charge of the team, and Agnes Jagers is the physician that closely works together with Dick to get this project going. Now, I want to change gears a little bit and talk about, again, BRCA2 and a new interactor that we have discovered of BRCA2 called HSF2BP. And HSF2BP stands for heat shock factor 2 binding protein. And we discovered it as an interactor of BRCA2, and it turns out that this protein is ectopically expressed in tumors and thereby interferes with homologous recombination that's mediated by BRCA2. And this can have implications for patients that ectopically express HSF2BP and their potential treatments that can be applied to them in the clinic. So BRCA2 is a large protein, has a number of proteins that interact with it. So many RAD51 proteins interact with BRCA2. And the function of BRCA2 is to load RAD51 on the recombination intermediate. Another famous interactor of BRCA2 is PELP-B2. And these proteins are important in homologous recombination, of repair of DNA double-strand breaks, but also of repair of stalled replication forks. And they cross over into a repair of another type of DNA lesion called a DNA interstrand crosslink. So DNA interstrand crosslinks are repaired by the Fanconi anemia pathway, but they also require these proteins of the homologous recombination pathway. And that's obvious because all the homologous recombination proteins have also been identified in the Fanconi anemia pathway and have these corresponding names over here. Now, in the past, we have shown that the BRCA2 protein is a heat labile protein. So if you heat cells to about 42 degrees, you reduce the amount of BRCA2 in cells tremendously. And we made use of that observation to find new interactors of BRCA2. We did that by making mouse embryonic stem cells in which the BRCA2 gene has a knock-in at the endogenous level that is labeled with GFP. And we use GFP, IP, SciLec mass spec experiments to find interactors. So the idea is that we compare 37 degrees to 42 degrees, and that should reduce the amount of BRCA2. And every protein that co-varies with the level of BRCA2 is a potential interactor. So here we see the results of such an experiment. So in this quadrant up here are the proteins that are less abundant at 42 degrees. So here's BRCA2, and we find PELP-B2 and RAD51, which we expect to find, but here's a new protein, HSF2BP. So we did some controls. We made knock-in cells for HSF2BP, for PELP-B2, for a control gene. And so we can do the reciprocal IP. If we IP with HSF2BP, we find BRCA2. And if we IP with PALP2, which is an interactor of BRCA2, we also find HSF2BP. So not much is known about HSF2BP. It's very conserved from fish to mammals. 20 years ago, there was a paper that said that HSF2BP is a testis-specific protein. So in order to get a clue for its function, we made HSF2BP knockout mice. And indeed, there's no HSF2BP in the testis of these mice. And they have a meiotic phenotype. So we have mislocalization of this RAD51 protein and DMC1, which is a meiotic-specific RAD51 protein, and they have defective chromosome synapses. However, the mice are otherwise have no overt phenotype. They are perfectly fine, maybe a little bit born in below Mendelian ratio, so maybe early in development, there may be a function of HSF2BP. Now, we did not find HSF2BP in most tissues except the testis, but we did find it expressed in a number, not all, but in a number of human cancer cells. And so that suggests that HSF2BP could be ectopically expressed and amplified in tumors. So we looked in public databases, and we found that there is indeed an overexpression of RNA transcripts of HSF2BP in a number of different cancers, for example, bladder cancer and gliomas. And we looked at the C-Portal bioportal database because it suggests that the HSF2BP promoter can be activated. And indeed, in about 1 to 2% of, for example, ovarian cancer, the HSF2BP gene is amplified. Now, in experiments that I do not have time to share with you, we looked at overexpression or ectopic expression of HSF2BP in cells, and that caused chromosomal aberrations that are also found in these Fanconi anemia mutant cells. And so because Fanconi anemia mutant cells are sensitive to interstrand DNA cross-linking agents, we made cells that ectopically express HSF2BP, and indeed those cells are sensitive to this interstrand cross-linking agents, just as Fanconi anemia patients, as if these cells have a problem repairing interstrand DNA crosslinks. Now, they are not completely homologous recombination deficient because the cells are not sensitive to ionizing radiation, which induces DNA breaks, double-strand breaks that are repaired by homologous recombination. And consistently, if we directly measure double-strand break-induced gene conversion, which is a recombination process in these cells, it's totally fine. So it seems the cells may be sensitive not to the repair of direct induced breaks, but indirectly induced breaks that are a consequence of stalled DNA replication, which is an intermediate and interstrand crosslink repair. And consistent with that, these cells that overexpress HSF2BP are also sensitive to the PARP inhibitor olaparib, which also causes replication stress. So we wanted to look at the interaction between HSF2BP and BRCA2 in some detail. And so BRCA2 is 3,418 amino acids, and we mapped it to a 68 amino acid fragment that is required for interaction with HSF2BP. And on the HSF2BP side, we found one point mutation here, arginine 200 to threonine, that is also unable to interact. We did some biochemical experiments that showed that purified HSF2BP and a fragment of BRCA2 can interact using a sizing column. So if you mix them together, the elution profile of the protein shifts from when they are separate. And that doesn't happen for the version of HSF2BP that does not interact. So we have done SEC-MALS experiments and find that the interaction is very tight, 1.2 nanomolar, and that HSF2BP is a tetramer, which interacts with 2 BRCA2 molecules. In order to prove that that is the interaction of HSF2BP with BRCA2 that causes the sensitivity to interstrand cross-linking agents, we made cells that have a version of BRCA2 in which exon 12, which contains the interaction domain with HSF2BP, is deleted. Those cells are totally fine. So BRCA2 can miss this exon for its function. And indeed, what we find now, if we ectopically express HSF2BP in cells that lack the interaction domain of HSF2BP on BRCA2, then those cells can no longer be sensitized to interstrand cross-linking agents or to PARP inhibitor, for example. So it's really the interaction between these two proteins that causes this phenotype. So we next wanted to understand what's the mechanism, how does it do it? And for that, we collaborated with Knipschier, who developed a biochemical assay based on Xenopus leavis extracts in which you can in vitro look at repair of DNA interstrand crosslinks. I will not go into the details, but in order to repair an interstrand DNA crosslink, there are a number of intermediate steps. So conversion of replication forks, unhooking of the crosslink, translesion synthesis over the crosslink, homologous recombination, and eventually a repaired product. We found that if you ectopically put HSF2BP in that extract, it is specifically the homologous recombination step that is inhibited. We did a sort of a chip experiment, and we found that if you add wild-type HSF2BP to these extracts, you no longer load the RAD51 protein on the DNA that has the interstrand crosslink. And that doesn't happen when you use the muted protein that can no longer interact with HSF2BP. To look more at the mechanism, we found that it's actually the BRCA2 protein that is progressively degraded during repair. So here we see that we plot for the amount of BRCA2 protein in the extract, and we see it disappears over time in the presence of wild-type HSF2BP, but not in the presence of the mutant version. We found that this degradation is proteasome mediated, that it absolutely depends on the interstrand crosslink. If the crosslink isn't there, the protein does not get degraded when you add HSF2BP. If you inhibit the proteasome, you still lose BRCA2 from the interstrand crosslink site. So even though the protein is not degraded, it's still lost from the interstrand crosslink site. And we found that that requires the P97 segregase. So the P97 segregase appears to remove BRCA2 from stalled replication forks so that it cannot load RAD51 protein and thereby inhibits homologous recombination. So here's sort of a visual summary of our model. At stalled replication forks, the presence of HSF2BP causes ubiquitination of BRCA2 and subsequent extraction by P97 and degradation by the proteasome. It also explains why homologous recombination per se is not affected, because the repair of plain old double-strand breaks as induced by ionizing radiation doesn't require a stalled replication fork. It doesn't go through that intermediate process. So those, even in the presence of HSF2BP, that aspect of homologous recombination is still fine. And so, of course, this finding suggests that patients with increased levels of HSF2BP, one of the therapeutic options that we should start thinking about would be treatment with either PARP inhibitor or interstrand DNA crosslinks. And with that, I will end with the acknowledgements of the people that did the second part of the work. It was driven mostly by a postdoc, Alex Zelensky, and it was a fine collaboration with Dr. Knipschier at the Utrecht Laboratory. So thank you for your attention.
Thank you, Professor Canard, for your wonderful presentation. We have time for one short question, I think. And one of the questions that was submitted is, how do you see the practical application of multi-omics analysis in oncology? Is this only in revealing the mechanism of action of disease, or also as a personalized diagnostics and or therapy monitoring?
Yeah, I think, of course, multi-omics has great promise in both those aspects, right? It has promise in prognosis, diagnosis, but in order to get there, you first need to understand mechanism, right? So this is where our research is mostly focused on trying to understand these mechanisms and implications of the presence of a certain marker or a certain protein. I definitely think that high throughput omics analysis has a place in the future of oncology for matching patients with the proper therapy, right? So we always, how I see the future of cancer research is that because we understand mechanism much more, which can be very different so the cancer that people have can be due to defects in different pathways and different mechanisms. And if we eventually have a drug or a tool for all these different pathways, then trying to find out what in one specific patient is defective is very, very important. And the only way to do this would be by proteomics, in a sense, supported by genomics. But genomics is always, of course, a correlation. And proteomics could be, there you get at the cause of things. So I think the combination of those would be powerful in combination with better understanding what's wrong in one specific patient. So great future for that.
Okay, very clear, thank you. Okay, then I would like to pass the word to my colleague, Wei Wu, who can then announce the next speaker.
Okay, thank you. It's my pleasure to introduce the next speaker. That is Clara van Gool. And Clara is professor of metabolic diseases at the Department of Pediatrics in RUMC. And Clara specializes in genetic metabolic diseases and mechanistic insights into new treatment options for children. And today we have the pleasure of hearing from her about inherited metabolic diseases and how to translate such big data sets into better outcomes for patients.
So our final speaker of today's symposium is Dr. Ahmed Mahfouz, assistant professor at the Human Genetics Department at the Leiden UMC. And Dr. Ahmed specializes in analyzing single-cell data. His lecture is entitled From Genetics to Neurobiology Through Transcriptomic Analysis. And I would like to give the floor to Dr. Ahmed.
Yeah, thanks a lot for the opportunity to present to you our work today. I would like to start by showing you this animation, which kind of summarizes what we think has happened in the past 20 years or so with the rise of identifying genetic variants in association to diseases in general and more specifically for brain disorders. There has been a lot of success from the genome-wide association studies or exome sequencing, where we come up with certain genes that have strong association to a certain phenotype. But nevertheless, most of these associations have very small effect sizes. And that has put a lot of burden on people who try to study more the biological aspects of these variants. Because generating, let's say, a mouse model, for instance, for a variant identified through GWAS that have a very small effect in general is not likely to yield something very, very useful. So the approach we take is a functional genomic approach, where we try to put in transcriptomic data in between these two ends of the spectrum, in the sense that we try to rely on gene expression patterns across different brain regions or across different development stages to guide us a little bit towards mechanisms or pathways that might explain the involvement of a specific gene into a certain disease or function. We heavily rely on resources that map expression across different brain regions and development stages, and these were mainly resources generated by the Allen Institute for Brain Sciences, where they took on this task of mapping gene expression in the mouse, in the human brain, but also in macaque, some years ago, using different technologies to measure mRNA abundance, such as in situ hybridization for mouse or microarrays and RNA sequencing in case of human brain. And based on that, we have developed a set of methods to basically help look into the data and summarize the information we have, whether that being mapping expression patterns in a way that makes it more accessible to people, providing tools to interact with the data, integration of data across multiple omics types, or looking at regulatory networks and interactions between genes. So today I want to present to you work that we published earlier this year, and this is work of former student Arlene Keogh, who looked at Parkinson's disease. And if you don't know, so Parkinson's disease is a progressive neurodegenerative disease. It starts with relatively mild symptoms, like a loss of smell, for instance, and then it develops the common phenotype in the disease with rigidity and tremors. And later on, patients also experience cognitive impairment and different forms of dementia. And these symptoms are also matched by different pathologies in the brain. So this has been staged by Braak. So if you're familiar with the Alzheimer's Braak stages, this is some sort of a similar staging system based on pathological evidence. And it does show that in pre-symptomatic stages like 1 and 2, there is pathology observed in the brainstem area, and then later on, at the time that the motor symptoms are observed, there is damage to the limbic system, and later on the pathology spreads to the cortical areas, and at that moment, patients do experience the cognitive impairment and dementia-like symptoms. And the question we had was that, can we actually find gene expression patterns that do map to these progression patterns across the different Braak stages? So for this, again, we rely on the Allen data. So if you're not familiar with this, this is a large atlas of gene expression across the human brain. So samples have been measured using microarrays, and some were measured using RNA sequencing. They come from 6 individuals, so a relatively small number, and I'll come back to that later. But the number of samples is still relatively large. So we're talking about 37,000 samples mapping across the whole brain. And each sample is associated with an MNI coordinate, as well as an anatomical annotation. So we know exactly where this sample comes from in the brain. So what we did is that we associated each sample to one of the Braak stages. So for instance, samples that fall at the brainstem part would be mapped to Braak stage region 1 or 2, while samples in the cortex will map to region 6. And you can see here which anatomical regions fall into these regions, and also the number of samples. So I'll not go into details, but just to mention that this high spatial resolution allows us to have actually a large number of samples in each stage. So what we do now is that we try to look for genes that we call Braak stage related genes. So these are genes that show a pattern across these regions, right? So they have high expression pattern, for instance, in region 1, and then this gradually decreases until you reach region 6, or the other way around. And for this, we rely on simple correlation analysis. So we correlate the expression of a gene with the order of the samples across these Braak stages. And we look also for genes that have a large difference, because we can easily find correlation, but the effect size is not as large between region 1 and 6, for instance. And if we do that, we actually end up with these numbers of genes that you see here. So we have 348 genes that have a decreasing expression pattern across the Braak stages, and we have 612 genes that have an increasing pattern across these Braak stages. Here I'm just showing you a summary of the expression of these genes. So if we look at the average expression of the genes across the different stages, and you see the multiple boxes here are representing the multiple donors. So as I said, the Allen data has 6 donors. So you see that the patterns are relatively consistent across these 6 donors. Of course, 6 is a very small number. So what we did is that we tried to validate these patterns in larger cohorts. So we looked at data from the UK Brain Expression Consortium and from GTEx, where we have relatively less samples. So you see here on the x-axis, we don't cover all the Braak stage related regions, but we do have a much larger number of donors than in the Allen data. So here 134 and 751 donors. And if you look at the average expression patterns of these genes across these regions, you see that basically we validate the patterns that we have picked in the Allen data, right? So it's not heavily biased by the 6 donors that we relied on. So genes for which we predicted a negative correlation, so high expression in early stages, low expression in later stages, tend to show the same pattern in these data sets, and the other way around. We also wanted to see what actually happens in PD patients. So we rely on data that was collected at the Amsterdam UMC. And this is data from patients that were graded at different BRAC stages, and these are the brain regions from which these samples were collected. So first of all, looking at the control samples in this data, you see that, okay, again, the same thing, that for genes for which we predicted a decreasing expression or an increasing expression, the control samples show the same pattern. But now if we look at the PD patients, you see this correlation pattern for these genes is totally lost, right? So we don't see the same pattern in the PD patients. Again, supporting the evidence that these genes are strongly associated to the progressive nature of the disease. What we also did is that we looked at genes that are known to be associated to PD through GWAS studies. So the ones that I show over here are either included in our list of negatively correlated BRAC-related genes or positively correlated BRAC-related genes. And perhaps I should comment on alpha-synuclein, for instance, and SNCA. So this is the gene encoding for the alpha-synuclein protein, which is the main form of the tangles or the pathology in PD. What you see here is that it is positively correlated to BRAC regions. So it has low expression in regions affected early and high expression in regions affected late, which is a bit in contrast to what we know from genetic studies. So from familial studies, we know that SNCA has a gain of function mutation. So the higher the expression of the gene, the more problematic it is. But at least based on expression patterns, we do see the opposite. And we do have a full discussion of this in the paper if you're more interested. So overall, I will not go through all the results from the paper, but what we do at the end is that we associate genes basically to specific stages of BRAC. So we know, for instance, if a gene has higher expression or low expression at a certain stage, and that might indicate whether the gene has a role in being in a region of the brain being more vulnerable or protected against the pathology that we observe. So one thing I want to point out is that we are comparing regions in the brain with very different cell type composition. So if we compare the brainstem to the cortex, for instance, we know that the cortex is much more rich in neurons compared to the brainstem. So it could very well be that the differences we're observing is just a reflection of this difference in cellular composition. So there are ways to check this using computational models. So we try to see whether the expression of neuronal markers, for instance, fully explains the differences that we see, which does not seem to be the case. So most of the results that we have got are independent of the cell type composition, but there are indeed limits to how much can we control for this given the bulk samples that we rely on. So I think this is a good moment to shift a bit to the single cell work that we're doing. So you're probably aware that for the last 10 years or so, there has been a lot of advances in single cell technology. And the brain, I think, is one of the organs or the systems that have been heavily studied with single cell technology, but has also benefited a lot from these studies. Indeed, one of the first large studies on single cell where we could actually profile thousands of cells was conducted in the brain from the lab of Sten Lennerson in Karolinska, where they looked at the somatosensory cortex and the hippocampus of a mouse. And since then, there has been a large increase in the number of studies employing single cell technology. So I think we're now at a stage where there's this exponential growth in the use of this technology. And if you look at only single cell RNA sequencing, you see that the brain is really one of the top organs that has been profiled with single cell RNA. So we kind of modify our approach a little bit. So instead of only relying on bulk gene expression data to bridge this gap, we'd also like to make use of a lot of the single cell data out there. There are, of course, a lot of single cell data specific questions that we would like to answer, but there are also a lot of challenges that this data poses to us. So the one I want to talk to you about today is this fact that we actually have a lot of multimodal single cell data. So perhaps single cell RNA sequencing is a bit more familiar, where you can take cells, take a tissue, dissociate the cells, and then you can basically sequence each individual cell. So this gives you a very a good overview of the whole transcriptome of the cell, as much as you can measure that with the technology. But you have a total loss of spatial information, right? It's a destructive technique, so we really have to dissociate the tissue. We have no information about where these cells come from. On the other hand, there are a lot of developments for spatial transcriptomics methods, where you can kind of scale up in situ hybridization approaches or single molecule FISH to get an idea about the RNA distribution across a tissue at a single cell resolution as well. So this allows you to retain spatial information, so you can look at the relations between cells, for instance, or the distribution of gene expression. But these approaches are in general limited to the number of genes or transcripts that you can assay at the same time. This was published last week, actually, and it's work from Tamim Abdelal, who's just finishing his PhD in the lab, where we present a method to actually integrate these two types of data. So the goal here is that we have a single cell RNA sequencing, where we have a profile of all the genes for a certain sample. And we have a matching spatial transcriptomic data from the same tissue. So it doesn't have to be exactly the same sample. And I'll show you examples that are not even from the same study. And typically in these methods, we have up to 1,000 genes that could be measured. There are protocols that can measure more, and we also discussed that in the paper if you're interested, but I'm not going to be talking about it. Our end goal here is that we want to actually predict the expression of genes that you have not measured in space, right? So what about the other genes, more than the 1,000 genes? How would they look like in the tissue? And we believe that with the help of a matching single cell RNA-seq data from the same tissue, we can actually make this prediction possible. I'll briefly explain the approach here. So we rely on a relatively simple method for domain adaptation called PRECISE, and this was developed by Sofiane Mouraghi from Delft and the NKI to basically translate predictions you can make on cell lines to human tumors. So the method is relatively simple. So we do a dimensionality reduction on both data types, and in this case we use PCA, but it's not restricted to principal component analysis. You can use other approaches as well. And then we do a singular value decomposition on the cosine similarity matrix. And this allows us to get these principal vectors, which are kind of similar to the principal components that we had earlier, but they are more aligned, right? So the first principal vector of the RNA-seq data would be similar to the first principal vector of the spatial data. And if we then project the spatial data on the single cell RNA-seq data, we basically have both data sets in the same space or the same domain, and we can do imputation of the measurements that we don't have in the spatial data. We rely on a simple KNN imputation, but again, this is something that's not really fixed in the method. So let me show you how does this look like. So we applied this to data from the primary visual cortex in the mouse. So we have StarMap data. So StarMap is a protocol to do spatial measurements. We have measurements for 1,500 cells and almost 1,000 genes. And from a totally different study, but still from the same region in the mouse brain, we have measurements for 15,000 cells, and this covers the whole transcriptome. So here I'm showing you an example where on the left column you have examples of genes, and this is the measured StarMap data. And on the right side, these are the predictions we made with SPAGE. So basically, we remove this gene from the data set, and then we try to predict the expression of the gene across the cell. And what you can see here is that the patterns largely map between our predictions and the measurements, even for genes that have a very sparse expression like BSG over here. So this looks quite good. So how do we actually compare to other methods? So here I'm showing you on the y-axis the correlation between our predictions and the measured values. And of course, these correlations are very, very low, and we can discuss this after the talk if you're interested. But if we compare how we perform compared to, say, RAT-Liger and GIMVI, which are comparable methods that can perform this task, you can see that it's significantly better. And this is a Bert t-test based p-value that you see. Now, we also came across examples where the measurements that we had from StarMap here on the left actually don't match our predictions, right? And we were a bit curious about this because the predictions are very specific, right? So for RORB, for instance, we do predict very strong expression in one layer of the cortex. So we wondered whether this was actually a mistake or something correct. So we looked at another data set, another spatial data measured using OSM fish. And this is from the visual cortex. So it's, sorry, I think this is from the somatosensory cortex. So it's a different cortical area, but you see that the expression of RORB is again restricted to the same cortical region as we predicted. So this does seem to actually be a wrong measurement from StarMap or a missed measurement from StarMap. And in that case, the prediction is actually correct. Again, the value lies in predicting genes for which we don't have measurements, but that makes it a bit difficult to validate. So these are two examples where we predict specific patterns of expression. And to validate these, we actually went back to the old Alain data. So this is in situ hybridization data from a totally different experiment. And if we zoom in on the same cortical regions where this tissue was measured, you see that the predicted patterns actually match what we know about the spatial expression patterns of these genes, which shows that the algorithm works quite well. The last thing I want to show you is that because the algorithm is relatively simple, right? We rely on very simple methods such as PCA and alignment of principal components. We actually think this is very scalable to large data sets, which are becoming more and more common. So we looked at the MERFISH data. So this is data from the pre-optic area in the hypothalamus. And here there were measurements of 65,000 cells across 155 genes. And in the same study, so this is really a matched piece of tissue, they also performed single-cell RNA sequencing. Again, similar picture in terms of performance. So SPAGE outperforms the other methods out there. But if we also look at the memory usage and the average computation time, here you see in black SPAGE compared to the other methods, and it's a lot faster, and it basically uses way less memory than the other methods. So we think this allows it to be scaled to much larger data sets that are more commonly generated now. So to summarize, we basically take this functional genomic approach to better understand the role of genetic variants in brain function and disease. I showed you one example where we used the Allen Brain Atlas to identify genes and pathways that are associated with the progression of Parkinson's disease. And in the second part, I presented to you SPAGE, which is a method to predict gene expression pattern at the single cell level. And I showed you that it outperforms the other approaches while at the same time being more scalable and flexible. And with this, I would like to thank the people involved. So for the first PD study, these are the people that were involved. And Arlen and Tamim are the main people who performed this work. Thank you.
Thank you, Dr. Ahmed, for this really interesting and challenging single cell spatial omics. I think we have time for 2 questions right now. One of the questions would be how other omics, and preferably, I guess, spatial omics technologies like protein expression and protein modifications, how they can help you, and would it be possible to incorporate that into your computational models as they are right now?
Yeah, so I did focus mainly on mRNA data for now. We do work a lot with protein measurements that are collected using cytometry data, for instance. And with cytometry, you can also do imaging mass cytometry, which allows you to, again, look at protein distributions, even at a subcellular level. Yeah, most of the methods that we develop are very general in the sense that we do handle different types of data. But yeah, there will be more on that in the future, of course, because we also have a lot more technologies that can do multiple measurements even in the individual cell. So doing mRNA and accessibility, or doing protein abundance and mRNA abundance. Yeah, so it's a lot of challenges, but I think it's also a lot of opportunities.
Great, thank you for your answer. Then I will pick just one question from the audience, and please do connect to Dr. Ahmed if you have further questions. But one of the things that also interested me is that the picture suggested that the spatial actually does a really good job in predicting. However, the correlation calculations appear to be rather low. How come?
Yeah, so this was also very puzzling for us at the beginning. I think actually correlation is not really the right measure to use here, but we're also very much at shortage of a better measure. So do let us know if you have a better idea. We did look at methods that look at mutual information or like the car index, for instance, Moran's eye, so we can kind of summarize the spatial patterns and what are the similarities between these patterns. One thing that does seem to be also very challenging is that we do predict the pattern, but not the exact expression value. Again, these methods are not very quantitative as well. So in a way, you can argue that we're more interested in the pattern rather than the exact expression number or how many molecules. But that would also be talking my way out of it a little bit. I think we can also do a bit more in the imputation side. So as I said, we try to keep things very simple. So we rely on this KNN imputation. And we actually show this a bit in the paper that even on single cell RNA-seq data, if you try to use this KNN-based imputation on the same data set, it does not perform very well. So this is also an area where we can improve. So yeah, so to summarize, I think we can improve the imputations a little bit, but we do also need a better measure to assess similarity.
Great. So thank you very much for the really interesting presentation and answers to the questions. And I would like to give the word back to Alain.
Yeah, thank you, Hans. And thank you, Ahmed, for, again, a very interesting presentation. And I think throughout the day, we had a great number of presentations. And it's now my job and honor, actually, to close it. So I think we had a wonderful day today. Where we had, again, a mix of nice science, technologies, different applications. And unfortunately, as mentioned, it's not a live meeting, but we did the second best. And I think it was pretty interesting to do so. So I'd like to thank all the speakers, actually, for their contribution today. I also like to thank all the sponsors, which I mentioned on this slide, for allowing us to do this meeting just free of charge, which hopefully we can do in the next years to come. And I'd like to thank them for the contributions. I promised that we should have an early glance on the group picture. And like I mentioned, it's kind of a Muppet show, which we organized today. So thanks to our back office team, we had this Muppet show view, which you can see on the slide here. We will obviously share this through our normal channels. And I think it was a good memory of a wonderful day we had. So in closing, I'd like to thank all the participants. And we had about 180 online today. Thanks for all the participation. I did mention also the Cross-omics community where we are sharing news and events and other interactions. So do join that. It's a LinkedIn group which has several news items during the weeks. And with that, I'd like to see you again at the third Cross-omics Festival. We now plan this in April next year. Hopefully it's going to be a physical meeting, but who knows what the status of the world will be at that. So thanks for your responses. And if you're done, just finish the Zoom and then we finish today.